Hey, so today's lesson we're going to be looking at biological molecules, specifically carbohydrates and lipids. So, just to draw your attention to the fact that I have these free interactive quizzes on my webpage, Biology by Bradford, that you can use at any point to review. So, they work, you could download them if you're a teacher and share them with your class, or you could print for a group. But the idea is that when you go through them, so why can carbon form such a huge diversity of biological molecules? You click on what you think is the correct answer, and you get a little bit of feedback as you go. So if you get one wrong, oh, accidentally got one right there, and then you get an explanation of why it's hydrolysis, for instance. And then you can reset, and what's linked to all of these as well. In the future, when there's an interactive lesson that goes with it too, that will also be linked. So today, we're going to think about, first of all, carbohydrates, and then after that, we'll talk about lipids. So when I talk about carbohydrates, I always start with thinking about CHON, CHONP. And I'll just move this one up. And if we can see here, CHO for carbohydrates, CHONP for lipids, CHON, and for proteins, and CHONP for nucleic acids. Got a bit tongue-tied there because of the P. But so the P and the S in the brackets, you might know, that's because they are found in some of them. So for example, with lipids, we sometimes, we have phospholipids. So there's sometimes phosphorus is needed, and it's needed in all of phospholipids. But the ones that are essential for any type of lipid is just carbon, hydrogen, and oxygen. So it's good to remember CHON, CHOMP to give you that basis of what elements are involved. Now, the monomers of a carbohydrate, so a single unit, is what we call a monosaccharide. An example would be glucose. It's a source of energy. And when we think about glucose, we've got two types of glucose, alpha glucose and beta glucose. A student pointed out to me once that he remembered alpha glucose as being alpha glucose, not beta, is because of only one of the OH groups, so the third one, is up and the other ones are down. And an alpha of a group is someone that dominates a group. So, for instance, here, like these are two what we call isomers. So, an isomer is the same chemical formula, so C6H12O6, but different chemical structure. And the structural difference is that first carbon here, the OH group on the beta is up, and then the alpha one is down. So these numbers here represent carbons 1, 2, 3, 4, 5, 6. And that's our structural backbone of these molecules. And then when we talk about bonds, like a 1,4 glycosidic bond, we mean it's between the first carbon of one and the fourth of the adjoining molecule. When we join two molecules together, so if we were to join these two together, we remove water. So that's a condensation reaction, or it's what we call an anabolic reaction. So Anna the builder is how I remember it, and breakdancing cat is the reverse. So breaking a bond is a catabolic reaction, and that would involve the addition of water. So it's hydrolysis. In this instance, if we were to join these two, it's a condensation reaction, anabolic, and you can also think like anabolic steroids. You've probably heard of bodybuilders using those to increase their muscle mass, so building muscle. And carbohydrates is one of those annoying topics where we just need to know some structural differences between different polysaccharides. So multiple sugars joined together is a polysaccharide. If it's two, it's a disaccharide. If it's a single one, it's a monosaccharide. So that saccharide is meaning sugar. Now we've got cellulose that we find in plants. That is a structural polysaccharide that helps with the rigidity of the cell wall of a plant. So we find cellulose in the cell walls of plants, and the OSE is a clue as well that this is a sugar and a carbohydrate as well. Remember, ASE would indicate that it's an enzyme. So a bond, 1,4 glycosidic bond, that means it's between the first and the fourth. And you can see here, that little line bit there, that's representing that chemical group at the top here, CH2OH. So we draw it like that. And because it's a 1,4 bond and it's a beta glucose, it causes the molecule to flip, alternating. and then we get a weak hydrogen bond between the chains. So that hydrogen bonding means that they are joined together with this like intermolecular force that holds that rigid straight chain structure. Now, we've then got two storage polysaccharides. So we've got starch and we've got glycogen. So starch is what we find in plants, and it's made up of amylose, which is spiraling. Something we just need to remember that it spirals. You might have to identify this molecule. I don't have a quick way of memorizing that. And we've got amylopectin. So yeah, amylopectin and amylose make up starch. Amylopectin is the branching one. So there's 1,4 and 1,6 glycosidic bonds there, which make it alternating with branching. You can see the 1,4 bond makes the straight chain, and the 1,6 is where it's branching off. So those two combined make up starch, which is the storage molecule for glucose in plants. And then the storage molecule in animals would be glycogen, found in the liver. We've got the hormones insulin and glucagon that we'll talk about in a homeostasis video that help regulate the concentration of glucose in your blood. And the way that it's stored when insulin is released is you're going to then take up glucose into your cells. And when it gets taken up into the liver, it's then converted into glycogen. So building this is going to involve a series of condensation reactions, which we know are anabolic. And so this is highly branching. The advantage of being branching is that enzymes can access the end of all of those chains to help break up the molecule in order to access that sugar. So part two, we're going to talk about lipids. I always start my class with this, thinking about this question. How can I remove the salt from my salted butter? Of course, you can pick up on my accent there when I say salt, but how do you remove it from your salted butter? So you can pause it, think about it if you want, and then I'm going to just tell you in a second. So you add hot water, and what's going to happen there is, of course, lipids are hydrophobic. They're going to separate from the water, but because it's hot, it melted the butter, it dissolved the salt that was contained within it, and then in order to get that salt, you would just evaporate off the water. But you leave it to all cool, and you've got your butters going to separate itself from the water because it's hydrophobic, being a lipid, a non-polar molecule. It repels from water. And then, of course, we can evaporate off the water to leave ourselves with the salt. So this is one thing that I super miss being living in Switzerland and coming from the UK is salted butter. It's the first thing I basically do when I move is to get myself some salty butter. Now, lipids are a bit simpler than carbohydrates because if we just jump back to this, there are no monomers. There's no true monomers. Here in this table, they've put like one glycerol and 3 fatty acids, but that's not a monomer. That's the components of a triglyceride. Did I say no monomers? There are no polymers. There are, of course, monomers. So sorry about that. Just sometimes when you're rambling, you go off on a tangent. But there's no polymers, but they're all made up of monomers. So the function is the long-term energy and storage within our bodies. Also, we've got like the phospholipids, which make up our cell membranes. So here we've got a triglyceride. So we have our glycerol and our 3 fatty acids, and then a phospholipid, which is in replacing one of those fatty acids with a phosphate group. And then we have steroids, and steroids are predominantly, hormones, which like cholesterol, estrogen, progesterone, testosterone. 3 of those there are hormones: estrogen, progesterone, testosterone that are going to help regulate processes in the body. And then cholesterol is something that wedges itself in the cell membrane. Or like one of its important roles in the body is to wedge itself in the cell membrane. And if you've watched the video about the cell membranes, and maybe it doesn't exist yet, but it's going to exist, then you will know that its role when it's hot is to kind of bring together the phospholipids, and the role when it's cold is to push apart the phospholipids. So it helps modulate the fluidity of a cell membrane. And a phospholipid here, these are what's making up our cell membranes in a bilayer. And when we think about lipids, the most important thing is whether or not they are what we call saturated. So there is no double bonds. That means it's straight. A trans unsaturated. These are man-made, and you can see that you've got the hydrogen either side of the double bond, and like in the cis one, I always think about it like this. The cis one, if I turn it on its side, it's a C, depending which way you look at. Yeah, there's my C. And that cis is a naturally occurring one and it's bent. And then that means that there's more space between any fatty acids. And that's why things like olive oil, which are polyunsaturated, contain loads of cis bonds within those fatty acids. That's why that they are liquid at room temperature. And they're good for you because they actually help get rid of the bad cholesterol in your body. And they help that cholesterol that they have helps like modulate the fluidity as well within our cell membranes. So, hope you enjoyed today's lesson, and don't forget to like, subscribe, and share. Check out the biological molecules little test as well to see how you can get on with that now. And of course, my other site that will really help you memorize those key terms is Cramly. And the idea here is it's space repetition. So all of those like jargon words that you think when I say them or when your biology teacher says them are like nonsensical, like anabolic, catabolic, glycogen, glucagon. How am I going to remember all of these words? Well, what works for me as a language learner, I'm learning French, and what will work for you as a student, and you are learning a language. When you learn biology, this is ultimately learning a new language. There's hundreds and hundreds of words within the HO and the SL syllabus. So space repetition is really going to help you lay the foundations. And this is my free study app. You can find it in the Play Store. You can use it on, you can download it as if you saved a home screen on the Apple phones, and you can also just use it on a desktop. But it's about doing a little bit every day, and then you really will see a big difference in your results in your tests. Okay, hope you enjoyed. Catch you later.